All right, so the first biomolecule we're going to discuss is carbohydrates. Carbohydrates are best known for their ability to store energy. However, they also serve a very important role in the structure of many organisms. Here we have a picture of a cactus. A plant uses cellulose in its cell walls to give them the structure and rigidity even when water is scarce. If we did not have cellulose in plant walls, so if you've ever noticed if you pick flowers or if you get vegetables and you leave them out, you cut them down and you leave them out for too long, if you don't put them in water right away, they'll start to wilt and get a little bit soft and mushy. Well, your flour will wilt, it might tip over, your celery might get rubbery, but it doesn't completely turn into a pile of soup, which is what would happen to you. If you had no bones, you would turn into a pile of soup. And this is because your cells do not have a thick cell wall made of cellulose. The celery relatively keeps its shape. The flour is going to keep its shape. It might tip over a bit, but it's going to keep its shape, maintain its shape and structure because of the cellulose that's found inside of the cell walls. It keeps them rigid even when water is scarce. We also have a crab right here. The interesting thing is there are actually a lot of animals that use carbohydrates for structure as well. So a crab is also pictured here because their shells are made out of another carbohydrate known as chitin. Mushrooms and funguses also use chitin as well. Bacterial cells here are going to be protected by a thick carbohydrate shell made out of a carbohydrate known as peptidoglycan. So carbohydrates can have not only an energy storage purpose, but also a structural purpose as well. Interesting thing to note about carbohydrates is that they contain carbon, hydrogen, and oxygen in ratios of 1 to 2 to 1. So whether we have a very, very large carbohydrate or a very, very small carbohydrate, for every carbon found in that molecule, there will be 1 oxygen and 2 hydrogens. Interesting thing about this, if you break this formula right here, what you have, if I cover up this half, you have a carbon and you've got H2O. Literally carbon water. This is what carbohydrate means. It literally means carbon water. Again, I'm not ever going to ask you to know that, but if it helps you remember the structure, to know that carbohydrates are found in a 1 to 2 to 1 ratio, 1 carbon for 2 hydrogens and 1 oxygen, if it helps you to know that carbohydrate literally means carbon and a water molecule, then there you go. But the chain itself can vary in length. Like I said, we can have very small carbohydrates, very long carbohydrates, but those ratios are always going to be there. Now, the monomer form of a carbohydrate is going to be what's called a monosaccharide. The polymer form is what's going to be known as a polysaccharide. Mono meaning one, poly meaning many. Here's an example. We're going to take just a quick look. Oh, sorry, there you go. The monomer is going to be the monosaccharide. The polymer is going to be what's known as a polysaccharide. Now we're going to take a moment to look at some of those monosaccharides. Here we've got it. Now there are many different types of monosaccharides, but the one that we're going to discuss most frequently in this in this class will be the glucose. Glucose is a monomer subunit for most carbohydrates. It is also what is known as a simple sugar. Monosaccharides and disaccharides are called simple sugars because they're relatively simple in structure. They're not made out of crazy long chains. They're just going to be those individual subunits. So a great example of this would be glucose. Glucose is a hexose. A hexose meaning that it is a 6-carbon chain, 1, 2, 3, 4, 5, 6. Anytime you see a ring structure like this, at every single apex, unless specified otherwise, we assume that it is a carbon. If you go into organic chemistry or even basic chemistry, you may see a lot of these structures. Unless specified otherwise, so over here, this corner of the hexose is being held together by an oxygen atom. In every other corner, if it is not specified, it is a carbon atom. Anyway, but what is something interesting about so here we have a 6-membered ring, but here, what do you notice about the functional groups? Here's a functional group, here's a functional group, here's a functional group, here's a functional group. What do you notice about these functional groups? They are all hydroxyl groups. What is the significance of attaching hydroxyl groups? Hydroxyl groups make the molecules polar, and when we have a polar molecule, that suddenly takes it from hydrophobic to hydrophilic. Glucose is very hydrophilic. Now, it won't dissolve in water. It's not going to break apart or suddenly ionize in water. It's not salt. It's not going to break apart in water, but it loves water. It very much likes water, which is amazing because glucose is going to be a major source of cellular fuel, and it's readily soluble in water. It can be transported throughout the body very rapidly because it can be transported in water. It can be taken up rapidly. It can be spread throughout the body rapidly because it is soluble in water. It is soluble in water because it is polar. It is polar because of these hydroxyl groups. See, it all comes back around. This is why we care about functional groups. Again, I'm never going to show you a picture of a molecule and say, hey, identify these functional groups and what they mean. But I do want you to understand and appreciate the significance of this, especially if you go on to take any other chemistry courses, or if you go into pharmaceuticals, or just in general. It's something you should be aware of, that this is very important that we take into consideration the solubility or the affinity that molecules have for water. So that's a monosaccharide. A disaccharide is going to be made up of two monosaccharides that have been joined together through, does anybody know what sort of reaction we use to join two monomers together? Hopefully you got it right. It is a dehydration synthesis. We're going to use a dehydration synthesis to join this monomer to this monomer. So there are a lot of disaccharides out there that exist in our everyday experience. Lactose, or milk sugar, is going to be a disaccharide. It's going to be composed of galactose and glucose. Lactose intolerant individuals lack the enzyme lactase, which breaks down lactose into galactose and glucose. So in other words, if you are lactose intolerant, it means that you do not have the ability to break down these covalent bonds that hold the lactose together. Sucrose is going to be composed of glucose and fructose. So sucrose, table sugar, is actually going to be made up of two different monomers, two different carbohydrate monomers strung together. Maltose is going to be composed of two glucose molecules. So there's a lot of disaccharides out there that are composed of multiple repeating subunits. So here I'm going to just show you a quick synthesis of maltose by using a dehydration reaction. All we're going to do is use a dehydration reaction. We're going to use two glucose molecules, two monosaccharides. We're going to kick off a hydroxyl group, kick off a hydrogen group, and so we've ultimately released water, and we now have maltose. We have a disaccharide. We took two monosaccharides, and we used a dehydration reaction to string them together to form a disaccharide and water. If we wanted to break that disaccharide down, if we wanted to break maltose down into glucose, we would just use a hydrolysis reaction. So we're going to start off with a disaccharide, we're going to add water to it, and we're going to break that molecule apart into two glucose molecules, each containing a hydroxyl group and a hydrogen group. So the sum total would be, this slide just kind of summarizes how we move forward with a dehydration reaction, or we can move backwards and break degradate that molecule using a hydrolysis reaction. So next up, I just kind of wanted to talk about, we've talked about sort of the structure or those synthesis reactions. The last thing I want to mention was what it looks like when we have a polysaccharide as a whole. Polysaccharides are going to be the polymer form of a carbohydrate. They're made up of long chains of monosaccharides that are all linked together. So this is our chain. Choo-choo, chugging into the station. And it's going to be composed of or made up of each one of these little cars all linked together. So these individual cars are going to be our glucose molecules, or glucose monomers. So polysaccharides are the polymer form of carbohydrates. They're made of long chains of monosaccharides all linked together. Because of their length and size, they're often going to be referred to as complex carbohydrates. Now, some of them serve the very useful purpose of short-term energy storage. When energy is needed in an organism, they only have to break down that starch. Just if we need glucose, we'll just snap right here and we've got a glucose molecule. If we need a lot of glucose, we'll just start cleaving all of these. We'll just chomp all these covalent bonds and release a whole bunch of glucose molecules. So that's fantastic, but we're going to store them in these great big long polymer chains that are wound really tight into this helical formation. And the reason we do that is because it serves two purposes. One, it conserves space, and it helps leave the sugar links exposed to hydrolytic enzymes for easy access to the sugar molecules. So we condense this all down, but if we need to access those covalent bonds and break them quickly to get at our glucose, we can do that. So that is our energy storage. Starch is how plants provide energy storage. They're going to have multiple forms, branched, which would be amylopectin, or unbranched, which would be amylose. I am not going to ask you to go into the details, but I do want you to know that starch is how plants store energy. Animals can also use polysaccharides to store energy. We can also use carbohydrates to store energy, but we do this in the form that's known as glycogen. Glycogen is how we humans store our carbohydrates, store our energy for short term as carbohydrates. And this is going to be found in the form of glycogen found in the liver. And the release of that sugar is going to be ultimately controlled by hormones that are going to be used. Insulin and parathyroid hormone are going to be responsible for releasing or uptaking glucose in our blood and storing it in the form of glycogen. Now, polysaccharides are ideal for energy storage because they're not quite as soluble in water. They're much larger than simple sugars. They are not going to pass easily out of the cell. They're not going to just sort of, be soluble in water and float right out of the cell. They're relatively easy to store and maintain. Now, there are also going to be structural molecules that are polysaccharides. So some are going to be formed as cellulose. Cellulose is pretty interesting. Cellulose is a really interesting molecule. So the cellulose is going to be found in the cell wall of plants. And you know what? We're going to come back to that in just a moment. So I'm going to skip forward to our next slide and we'll come back to that. But all right, so polysaccharides are going to be used primarily for energy storage. A polysaccharide is a polymer of monosaccharides. There are many examples. Starch is used for energy storage in plants. Glycogen is used for energy storage in animals. Now, there are some that are going to be some polysaccharides, some carbohydrates that have a structural feature. Cellulose, for example, is going to be how plants use carbohydrates for structural purposes. Chitin is how animals and funguses use carbohydrates for structure. Peptidoglycan is also a structural molecule found in the walls of bacteria. So I would like you to know this information, be familiar with the various types of carbohydrates, what their purpose is, whether they are used for energy storage or structure. Sorry, brain fart. Whether they're used for energy structure or energy storage or structure, and then whether they're found in plants, animals, funguses, or bacteria. This is a very great question. I love it. Usually appears in some form or another on a final. So please review those. One last thing I want to mention, cellulose. Cellulose is going to be the most abundant carbohydrate on Earth. Most abundant or organic molecule, really. There's over 100 billion tons of cellulose molecules are formed every single year. Unfortunately, we can't digest it. We can't break it down. Look at that. That is all cellular fuel. All of those glucose molecules are cellular fuel. We can't digest it, so we instead use it as what is known as a digestive fiber. So anytime you say or you hear, I need more fiber in my diet, what you are trying to get or obtain is cellulose. Cellulose is a carbohydrate that we can't digest. It's a structural carbohydrate, but it does help flush out our system and keep us regular because we cannot digest it. So the bulk of your waste is going to be molecules that you cannot digest, such as fiber and or cellulose. That's it for our discussion on carbohydrates. Next up, we're going to be talking about lipids.